All right, today we're diving deep into a process that is, quite literally, the engine of all biology, genetic mutation. Now, what are we talking about here? A mutation is any stable, heritable change in an organism's DNA sequence. These little alterations are the ultimate source of all genetic novelty. They're the reason bacteria develop antibiotic resistance, and they're the raw material for the incredible biodiversity we see all around us. We often think of them as mistakes or errors, but as you'll see, there's so much more than that. They're the very fuel for natural selection. Okay, so before we can talk about what goes wrong, we need a starting point, right? A perfect blueprint to compare everything against. In genetics, we call this the wild type. This DNA sequence you see here, that's our wild type for this explainer. Think of it as the standard, most common, fully functional version of a gene you'd find out in nature. We're going to use this exact sequence as our reference point to see just how different types of mutations can mess with the message it contains.
So what does happen when there's a typo? When the cell's copying machinery makes a mistake? Or when something from the outside comes in and damages this perfect blueprint? Well, that's when you get a mutant. And the consequences of these typos? Oh, they cover a huge range. A mutation could do absolutely nothing at all. Or it could give the microbe a brand new superpower. Or it could be totally catastrophic, shutting down an essential gene and leading to death.
So here's our game plan. First, we'll nail down exactly what a mutation is at the molecular level. Then, we'll look at where they come from. Are they spontaneous accidents or caused by outside forces? After that, we'll do a deep dive into the nitty-gritty of point mutations and the even wilder frameshift mutations. We'll unmask the agents of change, the mutagens, and finally, we'll connect all these tiny molecular events to the big picture, what it all means for microbes and their evolution. All right, let's get right into it and build our foundation. Before we can talk about the fallout, we need a really solid, precise definition. So, Section 1. What exactly is a genetic mutation? Okay, so the formal definition has two words that are absolutely key here: stable and heritable. Stable means the change has actually survived the cell's DNA repair crew. It's not just temporary damage, it's a permanent rewrite of the code. Heritable means it gets passed down to the next generation during cell division. Now, these changes can be tiny, micro-lesions affecting just a single base pair, which is what we're focusing on, or they can be huge macro-lesions, like deleting or flipping entire chunks of a chromosome, affecting tons of genes at once. Okay, we know what a mutation is. The next logical question is, where do they come from? It turns out they can pop up in two very different ways, which brings us to our second section, the difference between spontaneous and induced mutations. So on one hand, you've got spontaneous mutations. These are just the natural, unavoidable mistakes that happen inside a cell. You know, maybe the DNA copying enzyme, DNA polymerase, slips up and puts in the wrong letter. It's rare, but with trillions of letters to copy, it happens. Or maybe these weird little genetic elements called transposons decide to just jump from one spot in the genome to another, landing right in the middle of a gene. Then on the other hand, you have induced mutations. These are caused by an attack from the outside world, from an agent we call a mutagen. This could be radiation or a whole host of different chemicals that directly damage the DNA. All right, now for the really nitty-gritty stuff. For this next part, we're going to zoom in on the most common type of mutation out there, the microlesion. So welcome to Section 3, our deep dive into point mutations, where just a single base pair gets swapped out for another. Let's bring back our star player, the wild-type DNA sequence. Remember the central dogma? This DNA code is first transcribed into a messenger RNA, or mRNA. Then, a ribosome comes along and reads that mRNA message in little three-letter words called codons. Each codon tells the ribosome which amino acid to add to a growing chain. And voila, you get this specific chain of amino acids, which then folds up into a working protein. This is our perfect functional baseline. From here on out, every mutation we look at is going to mess with this process somehow. First up, and maybe the most surprising, is the silent mutation. This is where a base gets swapped in the DNA, but nothing happens. The final protein is exactly the same. How is that possible? Well, it's because the genetic code has some built-in redundancy. Think of it like having multiple words that mean the same thing. There are 64 possible codons, but only 20 amino acids. So several different codons can call for the exact same amino acid. And this slide just shows it perfectly. Look at the wild type. The DNA codon TTC gets transcribed to the mRNA codon UUC, which codes for the amino acid phenylalanine. Now look at the mutant. The DNA changes to TTT. That gets transcribed to UUU. But here's the magic. UUU also codes for phenylalanine, so even though the DNA is different, the protein product is identical. The genotype changed, but the phenotype didn't. Pretty cool, right? You know, the source material had a great way of putting this. It's like changing the sentence, "The cat ate the elk," to "The cat ate thy elk." Okay, yeah, the spelling is a little different, the information code has changed, but the meaning of the sentence? It's exactly the same. The message gets through perfectly. Okay, next up we have the missense mutation. And this is where things start to actually have an effect. In this case, the base swap changes the codon so that it now calls for a completely different amino acid. Let's look at the example. Our wild-type DNA has TGT, which codes for cysteine. A single letter swap to TGG now codes for tryptophan. Now, the impact of this swap can be anything from minor to major. If the new amino acid is chemically similar to the old one, say both are small and don't like water, it's a conservative mutation and might not affect the protein too much. But if you swap a tiny amino acid for a huge one or a positive one for a negative one, that's non-conservative and it could completely wreck the protein's ability to fold and function properly. And the analogy for this one? It's perfect. It's like changing "the cat ate the elk" to "the bat ate the elk." The sentence still makes sense grammatically, but the meaning has totally changed. A cat eating an elk is one thing, a bat eating an elk is something completely different. The severity of that change depends on the context, just like the effect of a missense mutation depends on the amino acids involved. And now, we get to what's pretty much always the most destructive type of substitution, the nonsense mutation. This one is almost guaranteed to be catastrophic for the protein. What happens here is that a base swap turns a normal amino acid codon into a stop codon.
Let's watch it happen. The wild-type codon TGG calls for tryptophan, but a single base change to TGA creates the mRNA codon UGA. And UGA doesn't call for an amino acid. It's a universal signal that just tells the ribosome, "Stop, we're done here." The ribosome just halts, releases the unfinished protein, and you're left with a short, chopped-off, or truncated polypeptide that is almost always completely useless.
And the analogy here just drives the point home. It's like changing "the cat ate the elk" to just "the cat ate." Full stop. Ate what? We have no idea. The action is incomplete, the full meaning is totally lost because the message was cut short. And that's exactly what happens to the protein.
You'd think a premature stop codon is as bad as it gets, right? Well, not necessarily. There's another class of mutation that can be even more chaotic because it doesn't just mess up one codon, it messes up every single codon after it. This brings us to the aptly named frameshift mutations. The whole key here is the reading frame. The ribosome reads the mRNA message in strict, three-letter chunks. A frameshift mutation is caused when you insert or delete one or two bases. It throws off the entire grouping from that point forward. It's like reading a sentence where every word is 3 letters long and then someone adds a random letter near the beginning. The rest of the sentence just becomes complete gibberish. This slide shows the absolute chaos this causes. Step 1, everything's normal. Step 2, a single G base gets inserted. Now look at step 3. The ribosome's whole reading frame is shifted over by 1. The old codons are gone. And the result, in step 4, is a sequence of amino acids that is totally wrong, completely nonsensical from the point of the insertion. And to make matters worse, you'll almost certainly hit a random stop codon pretty quickly, leading to a truncated protein filled with junky amino acids. It's a total mess.
So we've seen in detail what can happen to the DNA. Now let's talk about why it happens. What are the culprits behind these induced mutations? It's time to meet the mutagens, the physical and chemical agents of genetic change. We basically lump mutagens into 3 main categories. You've got chemical mutagens, which are molecules that can react with or impersonate your DNA bases. Then you've got physical mutagens, which are basically high-energy forces like radiation that physically break the DNA. And finally, you have biological mutagens, which are actually bits of DNA themselves that jump around and cause trouble.
Let's look at how these chemical mutagens work their dark magic. Base analogs are imposters. They look so much like a normal DNA base that the cell uses them by mistake, but they have a bad habit of pairing with the wrong partner, causing mutations down the line. Alkylating agents are vandals. They stick chemical groups onto the bases, changing their shape so they mispair during replication. And then you have intercalating agents. These guys are like molecular crowbars. They're flat molecules that wedge themselves between the DNA bases, distorting the helix and causing the replication machinery to slip, adding or deleting a base and causing a frame shift.
Physical mutagens use pure energy as a weapon. Think of UV radiation from the sun. It has enough energy to force two adjacent thymine bases to stick together, creating a bulky mess called a thymine dimer that blocks DNA replication. Then you have the heavy hitters, ionizing radiation like X-rays and gamma rays. This isn't subtle, this is brute force. It can literally smash right through the DNA backbone, causing double-strand breaks, which are incredibly dangerous and difficult for the cell to repair correctly.
And finally, we have these fascinating biological agents of chaos. Transposons, or jumping genes, are segments of DNA that can cut themselves out of one spot in the genome and paste themselves into another. When a transposon decides to land right in the middle of a perfectly good gene, it causes what we call insertional mutagenesis. This massive chunk of new DNA almost always completely destroys the gene's function. Okay, we've gone through the nitty-gritty molecular details of how mutations happen. For our last section, let's zoom way out. What does all of this actually mean for a microbe that's just trying to survive and make a living in the world? Well, the outcome for the microbe usually falls into one of three buckets. The most common result is a loss of function. The gene is broken, the protein it makes is useless. This is your typical outcome for nonsense or frame shift mutations. Then, much more rarely, you get a gain of function. This is the jackpot. The mutation actually gives the protein a new or improved ability. And finally, if the mutation hits a gene that's absolutely essential for life, you get a lethal mutation. Game over. And this brings us to one of the most important concepts in all of biology. You have to understand that mutation is a totally random process. A bacterium setting in a puddle of antibiotics doesn't try to become resistant. What really happens is that mutations are popping up all the time, randomly across the whole population, creating tons of variation. Most of these changes are useless or harmful. But just by pure chance, one random mutation might happen to make a bacterium resistant. Then, the antibiotic acts as a selective pressure. It kills all the susceptible bacteria, leaving only our lucky mutant to survive and take over. Mutation is random, selection is not.
So that leaves us with one final thought. We started this by calling mutation an error, a typo. And yeah, for one individual organism, it often is. But for a whole population facing a constantly changing world, that constant, low-level stream of mistakes isn't a bug. It's a feature. It's the engine of adaptation. It's the source of all innovation. It's the ultimate key to survival. So you have to ask yourself, is it possible that the ability to make mistakes is the single most successful strategy that life has ever invented? So that leaves us with one final thought. We started this by calling mutation an error, a typo. And yeah, for one individual organism, it often is. But for a whole population facing a constantly changing world, that constant, low-level stream of mistakes isn't a bug. It's a feature. It's the engine of adaptation. It's the source of all innovation. It's the ultimate key to survival. So you have to ask yourself, is it possible that the ability to make mistakes is the single most successful strategy that life has ever invented? So that leaves us with one final thought. We started this by calling mutation an error, a typo. And yeah, for one individual organism, it often is. But for a whole population facing a constantly changing world, that constant, low-level stream of mistakes isn't a bug. It's a feature. It's the engine of adaptation. It's the source of all innovation. It's the ultimate key to survival. So you have to ask yourself, is it possible that the ability to make mistakes is the single most successful strategy that life has ever invented? So that leaves us with one final thought. We started this by calling mutation an error, a typo. And yeah, for one individual organism, it often is. But for a whole population facing a constantly changing world, that constant, low-level stream of mistakes isn't a bug. It's a feature. It's the engine of adaptation. It's the source of all innovation. It's the ultimate key to survival. So you have to ask yourself, is it possible that the ability to make mistakes is the single most successful strategy that life has ever invented?